Hi, my name is Anne Pringle. I'm a professor in the departments of botany and bacteriology at the University of Wisconsin-Madison. And in this third of the 3 talks I'm giving for iBiology, I'm going to dig a little deeper into the concept of convergent interactions and think about what an understanding of genome architectures of symbiotic fungi can teach us about this idea. Before I go any further, I want to acknowledge my 2 co-authors in this talk, Leonora Bittleston, standing here in the snow, and Jacqueline Hess, taking shelter underneath the mushroom. The work that I'm going to talk to you about is primarily thought through and driven by their PhD and postdoctoral research, respectively. So a long time ago, when Darwin wrote The Origin of Species, he recognized that there can be species that are unrelated to each other, but that look very similar. He called that analogical resemblances. We call that convergent evolution. So if you look at the photograph of this manatee and the beluga whale underneath it, you can see that they have a similar shape. And it's probably because they're subject to similar kinds of selective pressures in their different habitats. In fact, it's almost certainly because that's what's going on. So they're unrelated, but they look alike because of these common selective pressures. There's been a convergence towards the evolution of a similar form. We have been thinking about that idea of convergent evolution in the context of a very different kind of biology, if you will, an interaction between 2 organisms, not just the shape of a single organism by itself. What you're looking at here is a photograph on this book cover of seedlings of plants, pine seedlings. When you look in the soil matrix, you do see plant roots, but most of what you're looking at here are networks of hyphae. These are fungi, the bodies of fungi. And these networks of fungi scavenge in the soil. They go to places where plant roots can't go. They find scarce resources like nitrogen. They bring that nitrogen to the plant in exchange for carbon. So it's a classic exchange, benefit for benefit, trading of resources. This is the mycorrhizal symbiosis. In a mycorrhizal symbiosis, if you were to study it, one of the things that you would see is this very characteristic structure called a root tip. So what you're looking at is the tip of a plant, and it's encased or ensheathed by fungal tissue. When you see a root tip, if you cut it in half and you stain it, you'll see these very characteristic, again, network-like structures that are called Hartig nets, after the person who described and discovered them. When you see root tips, when you cut them open, you see Hartig nets, you know what's going on. You know that there's an exchange of resources between a fungus and a plant. This is a very clear morphological diagnostic of the interaction. And even if you don't know anything else about the species involved, you know that this is a mycorrhizal symbiosis. But it turns out that this interaction has evolved repeatedly, over and over again. You're looking at a large phylogeny or tree of relationships among different species, and anytime you see bold, that marks the origin of an ectomycorrhizal symbiosis, a mycorrhizal symbiosis. So you can see that it hasn't happened just once in nature. It's happened a lot. The interaction itself has evolved repeatedly in nature with this characteristic diagnostic morphology. So, for example, North American pines and mushrooms in the genus Amanita evolved the mycorrhizal association with that characteristic Hartig net independently of the evolution of Australian Nothofagus trees and Australian truffles. It's a really intriguing phenomenon that we have observed for a long time, but haven't really given a name to in quite the way that we can name it when we use the word convergent interactions. Convergent interactions can also involve communities, not just these pairwise interactions. So, for example, cows, colobus monkeys, kangaroos, and the Watson bird that you can see here all have foreguts that ferment the food that they eat. And it turns out that their gut bacterial communities also have a convergent structure and function. So we've defined convergent interactions as the independent emergence of multi-species interactions with similar physiological or ecological functions. It's a new way to think about something that I think we've known about for a long time, that there are interactions in nature that we can see that evolve repeatedly. In trying to think more about what it means to use these words, convergent interactions, we wanted to turn to a different system and really test what we were thinking about when we used these words. And the system that we turned to first is a system that involves two genera of what are classically convergently evolved forms. The pitcher form of Sarracenia, which is the North American pitcher plant, and the independently evolved similar form of the pitcher plants of Southeast Asia in the genus Nepenthes. Sarracenia and Nepenthes are very distantly related. They're not at all the same kind of plant in terms of what their ancestors are, but they look remarkably alike, as you can see in these drawings. They both form pitchers which house pools of water inside them. So why does a pitcher plant have a pitcher? Why does it have these pools of water inside it? A pitcher is basically a habitat sitting out on the landscape with this water inside it, and things fall inside the pitcher. Insects fall inside the pitcher, and they're eaten. It's a way for plants to get access to scarce resources when they're living in habitats that don't have that have very poor soils, for example. So that's what they're doing. They're eating insects in order to gain nutrition, and the Nepenthes do it in Southeast Asia, and the Sarracenia do it in North America. It turns out that these pitchers, when they first form and they're closed, are sterile. So it's only after they open that they begin to have other kinds of organisms fall inside them. And it's not just insects that fall inside pitchers. In fact, whole communities of bacteria and fungi and other kinds of basal eukaryotes are all found in this microcosm, this miniature ecosystem that the pitcher forms after it opens. So one way to think about it is that these plants have very conveniently placed little test tubes all across the landscape. And here you're looking at a picture of Leonora in a North American Sarracenia habitat. And you can see all the different kinds of pitchers that are scattered across this very beautiful habitat. So to test the idea of, so these pitchers are convergently evolved. They look like each other. What about their interactions with all of those organisms that fall inside them? Is there something convergent about those ecosystems that form in Southeast Asia inside Nepenthes versus the ecosystems that form in Sarracenia in North America? And to answer that question, we did a lot of different kinds of experiments, but the one that I'm going to tell you about today, we took Southeast Asian pitcher plants, Nepenthes, and we put them into a Sarracenia habitat to see what would happen. First, I'm just going to show you data, and this is the kind of slide that took Leonora 5 years to put together, and I'm going to show it to you in just a moment, but just to acknowledge that took a lot of time for her to go all across Southeast Asia and North America, collect pitchers, sample inside them, do a lot of DNA sequencing of entire communities inside those pitchers, and then use a technique called ordination to put every sample on a plot like this. And the interpretation is that if 2 points are very close together, they're closely related. And each of these points each point represents a single pitcher that she sampled. If 2 points are very far away from each other, then they're not closely related. So in this data set, what you're looking at are natural samples, not yet the experiment I'm going to focus on. In these natural samples, you can see that the Nepenthes species are different from the Sarracenia species in terms of their bacterial composition and the composition of eukaryotes inside them. But each is very distinct from the communities that you can find in the surrounding soil or in the surrounding bog. So often these plants are growing in quite boggy habitats. While the Sarracenia and Nepenthes seem to cluster a little bit apart from each other, if you look at the data in a different way, what you see is that they're not exactly the same kinds of organisms in the Nepenthes and the Sarracenia, but they're closely related to each other as compared to the organisms that you see in the surrounding community. So to illustrate that, this is a massive phylogeny, almost too dense to show you in a single image, of all of the organisms that we found in either Nepenthes, Sarracenia, or surrounding bog water or soil. The outer ring, the brown, are organisms that we find in bog water or soil. What you can see is that quite often, when you're finding subsets of the diversity that are found in Nepenthes, those same subsets of diversity, again, not exactly the same species, but very closely related species, are found in both the Nepenthes and the Sarracenia. So it's as if you found in Nepenthes and Sarracenia, in one you found a chimpanzee, and the other you found some other kind of monkey. So they're not the same, but they're closely related. And in the soil you found a beluga whale. So they're just the pattern that emerges is quite intriguing in terms of the phylogenetic relatedness. And by the way, why would you expect to find the exact same kinds of bacteria in Southeast Asia versus North America? So instead, you become more interested in the function in those closely related subsets of organisms that you find in those different places. Back to the experiment. So for this experiment, we planted in a bog Sarracenia purpurea, that's the native pitcher plant to North America, the same species in pots, multiple Nepenthes species in pots. By the way, we were very careful about how we did this work, where we got the plant material, how we imported it, and how we put it out in the landscape, being mindful that we didn't want to introduce non-native microbes into the local microbial community. We also added sterile glass tubes that either had or did not have insect prey, in this case, dead ants. So this is the experiment, and it's really about the most radical test of convergent interactions you could probably imagine. So we put these convergent forms together. You can see the glass tube, you can see the Sarracenia, and you can see the Nepenthes all together here in a common, in a shared space in the bog, and we replicated this across the bog. And the question is, does any kind of pitcher form assemble a Sarracenia purpurea community? So these are the results. So the first thing, and again, each point represents a pitcher, and the community, the diversity, in this case of bacteria, inside that pitcher. So in red circles are the Nepenthes from Southeast Asia, the data that I showed you before. And in orange are the Nepenthes from the experiment that I just described, the Southeast Asian Nepenthes from the experiment from the North American bog, the Sarracenia, and gray are the test tubes. If it's a triangle, that means the pH in that microcosm was less than 4. And the first clear signal we got out of this experiment is that pH drives the bacterial communities. So if the pH is less than 4, those samples cluster away from other samples. But in the experiment, the Nepenthes of that experiment and the Sarracenia of that experiment cluster together. You can see the yellow and blue intermingled here. And interestingly, the communities that formed inside the test tubes, which is the same kind of shape, a pitcher shape, cluster somewhat away from the Sarracenia and the Nepenthes. And similar patterns were found in the eukaryotic community, except that it wasn't driven by pH. So the relocated Nepenthes do seem to acquire Sarracenia-like communities when acidity levels are similar, was the takeaway that we got out of that experiment. So convergent interactions really seem to be a phenomenon in nature. it does seem that you have certain kinds of interactions that evolve over and over again. And by the way, I haven't said much about function, but it's not because we're not thinking about it. And we are thinking about how to understand what the functions are of these microbial communities inside the different pictures. With a very different system, then, we started to think about why it might be that you have these kinds of interactions evolve repeatedly. And the system that we are using to think about that aspect of the problem are these ectomycorrhizal fungi. And I'm going to talk to you now about a genus that's near and dear to my work, and that's the genus Amanita, typified here by this very beautiful Amanita muscaria. It's a very common, charismatic, and I think well-known fungus that you might find, for example, in any book of fairy tales. And this is an ectomycorrhizal fungus. So if I were to dig underneath this mushroom, I would find those characteristic root tips. If I cut those root tips, I would find heartig nuts. And this mushroom is quite likely forming a mycorrhizal association with the tree that you can see behind it, just the base of that tree. I first began work with Amanita because I was fascinated by a different Amanita species, the death cap, which you can see pictured here. This is Amanita phalloides, which is invasive in California. Because I had become fascinated by the problem of invasion biology of mycorrhizal fungi and started work with the death cap, a lot of things in my lab began to focus on the genus and what do we know about the genus and how it's distributed and what its evolution is. And a graduate student in my lab, Ben Wolf, became really interested in the problem of the origin of the ectomycorrhizal symbiosis within this particular genus. And it's another slide that took many years to put together. He generated this phylogeny. What you're looking at are lots of different Amanita species. And what he discovered is that there are all these species of Amanita that are ectomycorrhizal. And he discovered that they grouped together and away from this group of Amanita that are not ectomycorrhizal. These are free-living species that decompose probably mostly grass litter. We don't know enough about their natural history. But they're decomposers. They don't form these symbioses, whereas all these species do. So it lends itself in a really nice way to understanding the genetics of the ectomycorrhizal symbiosis because we have these closely related species with these contrasting ecological niches. So if we want to start to understand why this ectomycorrhizal association has evolved over and over again repeatedly, perhaps digging into this genus and the genetics of the ectomycorrhizal symbiosis here will give us some insights as to what's going on. What has been known now for a few years is that after the evolution of an ectomycorrhizal symbiosis, those ectomycorrhizal fungi lose the genes that would normally enable decomposition. So this is a large phylogeny that illustrates that point for many different genera of fungi, but I'm going to zoom in just to show you the data for muscaria, that's Amanita muscaria, and Amanita tiersii, which is an Amanita I haven't talked about yet, but is a decomposer. And you can see from the size of the circles that the circles are bigger for tiersii, which is a decomposer, and those circles represent numbers of decomposition genes. And so it's just what I said. The Amanita muscaria, ectomycorrhizal with that tree I showed you, has fewer decomposition genes than the tiersii. So we know something, but this loss, at least we thought at the time, doesn't really tell us very much about the genes involved to make something symbiosis. So it's still somewhat a mystery. How do you get to being a symbiont? What does that teach us about convergence? So in a thought that we would be able to answer this, thinking that we might be able to answer this question if we dug a little bit deeper into this genus, we sequenced 6 genomes, 3 ectomycorrhizal genomes, and 3 genomes from species that are not ectomycorrhizal, that are free-living. So what did we do with these genomes to try to understand the ectomycorrhizal symbiosis? Well, what we did first was the most straightforward job, was just to compare and contrast the different genomes to each other. So in previous slides, in this slide, and in future slides, green always marks the symbiotic fungi that associate with plants. Black are the fungi that are free-living decomposers and do not associate with plants. So these are just some basic genome statistics. And what you can see right away is that there are 2 ectomycorrhizal species with large genomes and one that does not have a large genome. By and large, the non-ectomycorrhizal species have smaller genomes, but there really isn't a clear pattern 3 versus 3. If you look at the predicted genes, again, there's not a particularly clear pattern that distinguishes the 3 symbiotic ectomycorrhizal species from the 3 species that are free-living and decomposers. So there's nothing striking right away that says this is the hallmark of an ectomycorrhizal fungus. And by the way, this statistic is just to tell you that we did a good job with the genomes, and we had a pretty complete data set by the time we were done collecting the data and analyzing them with our bioinformatics pipelines. So now let's dig a little bit deeper into what are the commonalities or differences between these 3 ectomycorrhizal species. So one thing that's true is that the genome expansions that you see in brunescens and polypyramus seem to be supported by seem to be caused or driven by expansions in transposable element content. So you can see, for example, that these bars, which mark the numbers and kinds of different transposable elements, you can see that they're quite large for brunescens and polypyramus. But again, it's not universally true that the ectomycorrhizal species are different from the decomposer species in any consistent way. As you can see, Tiersii, which is a decomposer, has more transposable elements than Muscaria. And by the way, the reason you see 2 Muscarias listed here is we actually sequenced the genome twice, just to test originally whether we were doing a good job and could replicate our data independently in a different laboratory. At the end of this, it seems like at least at the moment, there doesn't seem to be a thing that uniquely identifies ectomycorrhizal fungi as different from the decomposers. And in fact, it seems like distinct evolutionary processes are shaping each of the ectomycorrhizal species, as we saw from the data on genome size, protein coding genes, and the transposable element data as well. What about the amplification of these 2 in genome size of these 2 ectomycorrhizal species? How did that work in evolution? There are basically 2 hypotheses. One hypothesis is at the base of the ectomycorrhizal phylogeny here, there was a large-scale amplification in genome size, and then over time that was lost in one of the lineages. That's hypothesis 1. Hypothesis 2 is that there was the independent amplification of genome size in these 2 different ectomycorrhizal species. And if you model that process, what you find is support for the second hypothesis. If you do an analysis of where genes first appear, it seems that there is a lot more duplication independently in these species versus at the origin of the ectomycorrhizal symbiosis. And in fact, if you do a profile of the duplication of the gene families, here we've numbered the different nodes. You can see that it's node 3 and node 5. Follow them down and see the dots, and then follow back to see numbers of duplicated gene families. It's those 2 points on the phylogeny that have the most duplications. Again, supporting the second idea of an independent amplification of genome size in the 2 places. Another way then to think about these data is, okay, well, we still have 3 species that are ectomycorrhizal and 3 species that aren't. What gene families are only found in the ectomycorrhizal lineages? And here what you're looking at is sets of gene families for the different species, and I've overlapped them in a way so you can see what's shared or not shared. So for example, Polypyramus shares 27 gene families uniquely with Tiersii. And if you look to see where the three ectomycorrhizal species, Muscaria, Polyperamus, and brunescens, overlap, there's only 102 gene families that are uniquely ectomycorrhizal, which in the context of everything you're looking at is not so much. You might have thought that such a complicated structure as a root tip and a hard egg net and such a seemingly complex interaction as the exchange of benefits between a plant and a fungus might have involved a lot more. But within those 102 gene families, there are some intriguing signals, not least of which is that oxidative metabolism seems critical. These genes that are responsible for oxidative metabolism seem to be at play in the origin of the ectomycorrhizal symbiosis, and that's interesting because those kinds of genes are the same genes that are often implicated in the detoxification of plant compounds and signaling and other kinds of interactions. So I've shown you that genome architectures of the different species of Amanita are diverse, and that the data support one hypothesis over the other, the independent amplification of genome size in two lineages. At the end of these experiments and analyses, it seems that early EM evolution really is dominated by the loss of gene families, those decomposition genes. And there seems to be comparatively little gain of function at that origin. So how does this relate to the idea of convergent interactions and the pitcher plant work and the concepts that I presented at the start of this talk? Well, remember that one of the things that I showed you was that this interaction, this ectomycorrhizal interaction, has evolved repeatedly over and over again, which just seems remarkable. But maybe they have evolved over and over again because this dynamic of the massive shedding of genes with relatively few gains is a relatively easier evolutionary dynamic than the gain of lots of different kinds of things. Maybe losing things is always easier than gaining things, and maybe that's why these things have evolved repeatedly. And with that, I'd just like to thank Leonora and Jackie once again, and also thank the funding sources that have supported this work through the years. Thanks very much for listening.